Hyvää iltaa. Tervetuloa tänne luonnonfilosofian seuran tiistai-illan luentotilaisuuteen. Dear participants, welcome to this evening lecture of natural philosophy. Finnish Society for Natural Philosophy. Today we have as a speaker Professor Leonardo Almeida-Souza. He works in cell biology in Helsinki Institute of Life Sciences. He comes from Brazil, and that's why he also speaks English now. The theme is originally DNA, technology, principles, history and uses, but has the subtitle DNA, the secret of life. Professor Almeida-Souza, please.
Okay. Thank you. Can you hear me? Is that okay? So first of all, thank you for the opportunity to come and talk about the subject of my life, the only common denominator of everything I have done in the last 25 years, which is DNA. I have gone through many different fields: plant biology, microbiology, cell biology, human genetics, and etc. And DNA and recombinant DNA has been the only common denominator among that. So I'm really happy to be here and to talk to you a bit about this incredible molecule. And to do that, I will give this talk a bit of a mixture of history of the field. So I think the way we can understand why we do things the way we do today, it's by understanding why history led us to do that. And I will try to give, as I'm not going to be so technical, because I think that's not going to be very helpful. I'm going to try to talk about the history. You guys are going to understand why DNA has become something so interesting today. And at the end, I think that's going to be the interesting part. I'm going to discuss about the latest developments in DNA technology and how that has had a massive impact when we think about what we humans can do to ourselves in the form of genetic engineering. So, as I said, we're going to have a bit of history, travel through history a bit here. And we should start in 1953. The place is England, and in a regular pub in a typical academic city in England, where academics go to have lunch every day, there was a day in February where two of those academics stood up and said, "Ladies and gentlemen, we have discovered the secret of life." Not very small words, but they did it. Nobody knows exactly what happens after that on the pub, but we know what after that in science, and that was a big revolution. So the pub was the Eagle in Cambridge, UK. It's a pretty nice pub, and nothing special about it, but it's a decently common pub, English pub. And the gentlemen are Francis Crick and James Watson, and they have found out how the DNA molecule looks like. And that is a very interesting finding, because they literally got a lot of data from a bunch of people, and they got pieces of molecules of DNA made of metal, and start to figure out how they can come together with each other based on the data that was available, and they came up with something that was absolutely correct and absolutely revolutionary. So, and that story that I just told you, it's on a little plaque on the pub in Cambridge here that says the discovery of DNA, when they claimed that we have discovered the secret of life. I had a few pubs there. I had a few pints in that pub. That's me, close to the place, to the plaque there. And here is one interesting story about science that Francis Crick and James Watson and Maurice Wilkins got all the credit for that discovery. And they left behind a lady who had done an incredible job called Rosalind Franklin. And I am very happy that when I stayed in Cambridge for 7 years, and just below that plaque, they added a little plaque to give credit to the incredible work of Rosalind Franklin, whose results were really crucial for Watson and Crick to discover the structure of DNA. And what makes their discovery so incredible is the simplicity of it. This is the full paper from Watson and Crick that gave them a Nobel Prize 7 years later. It's really not very large, has 6 references. It's a tiny little thing, has not even a single experiment and 1 figure where they show what they have found. So we're going to come back to that a bit later on, because before trying to discussing the findings of Crick and what they mean and what they triggered in the scientific community, we have to travel almost 100 years and talk about a monk in a monastery in the Czech Republic called Gregor Mendel. And Gregor Mendel was not a typical monk because he had a lot of scientific interests, and he had a very thorough scientific program running in the monastery. And he had one question, which was, how do traits are passed through generations? At that time, in 1860s, people thought that the way that characteristics are passed from parents to kids, it's like mixing up 2 liquids in a cup. These things mix up and they just become impossible to put them apart. So effectively, we're just mixing up various liquids one after the other, and that would be how heredity would work and characteristics would pass. And Mendel decided to test that. And to do that, he was working with peas, and he had peas in his garden that he was doing very careful crossings, and those peas had different characteristics. Here are just the typical characteristics of peas. And I'm going to just give you an example of what he has done. He's done multiple of those. So he had a set of peas which were green, and if you would cross those ones with each other, they would always be green. And there was another set of peas which were yellow, and if you would cross them with each other, they would only be yellow. And when they would cross the green with the yellow, all the progeny of that cross would be green, always. And if you cross this progeny together, you would always have the next progeny on a very clear frequency of 3 greens to 1 yellow. A result that at first sight, I can imagine how difficult it was him to figure this out. And he came with a very beautiful explanation and a nomenclature that we kind of use until today. And this is his explanation, that the green trait, the green gene, was a dominant gene, and he gave a capital letter to it. So the G, the large G, the capital G is a dominant gene. And the green plants, the initial ones, had double Gs. The yellow one had the recessive gene. So that one did not have the same power as the dominant gene, as the name says. And if you cross those together, all the second generation will be large G, small g, because the big G is dominant, makes all the plants green. And if you cross those ones to each other, you have all this nice segregation here, which explains beautifully the 3-to-1 ratio that he sees on the progeny. He did that for a bunch of traits, and this is true until today. It's beautiful. However, biology is a bit more complicated. And not all things in biology are just one gene with a dominant and a recessive allele. So there is a lot of what we call non-Mendelian inheritance, where, for example, the 2 Gs the big and the small g, they are co-dominant. And there are cases where you do not have G big and G small, you have G, G1, G2, G3, G4, G5, G10, G100, so you have multiple alleles for the same gene. And multiple other traits, you need 5, 10, 20 different genes to give the characteristic. Nonetheless, the principle was right. And Mendel gave the name of, actually I forgot to say, elementin, which is a particle that is given to you by the mother and the father, and that particle can after that be dissociated. So that whole idea of two liquids mixing up in a cup was down the drain. There was an elementin, there was a particle, there was something in the cells that was being segregated, right? So we then go from 1862 to 1928. From that moment onwards, the timelines are a bit more fuzzy, so you have to be sorry here. And the question at that time, what people were asking was, so we have these elementin, what are they? At that time, people knew what cells were composed of. We didn't know about sugars, lipids, DNA, RNA, and so on and so forth. So what is that elementin? What is the chemical nature of heredity? And the answer came from a very unexpected place. A guy called Frederick Griffith was studying Streptococcus pneumoniae, which is a bacteria that causes pneumonia, and he could study that in mice. And when you have this bacteria, you have two types of them. One, which is called the rough strain, and that strain is non-virulent. In other words, it does not cause a disease, so you can inject that strain on the mouse, and the mouse lives, no problem. And you have another type of strain, which is called the smooth strain, and that strain is virulent. You inject on a mouse, and the mouse dies. And he did a very interesting experiment trying to figure out what was going on there, that he got this virulent strain and he boiled it. Boiled it so it killed the bacteria, but the components were still there. And he injected in the mouse, and the mouse was leaving. So he said, okay, there's something a bit more complicated. But the real unexpected experiment was when he got the boiled strain, the heat inactivated strain, and combined it with the non-virulent strain. And the mouse died. So something from the virulent strain was passing to the non-virulent strain and making that strain lethal and killing the mouse. And that's when he defined what we call today the Griffith's transforming principle. There was something that was being transferred between one bacteria and the other and giving them the virulence, right? A few years later, three other scientists called Avery, McLeod, and McCarthy, they took the experiments from Griffith and said, let's take it to a next level. Let's get that boiled bacteria that had the transforming principle, and let's try to figure out what is that. And they did a very elegant experiment. By the way, I'm just going to say this is a massive simplification of their experiment, because what they did at that time, they did not have the tools we have today. They have gone through hell to be able to prove that. So I'm giving a very simplistic view of what happened here. So they had the heat-killed bacteria. They could remove lipids and carbohydrates from that soup of boiled bacteria. And they got that, and they got that and spread it into a few tubes. In one of them, they added a proteinase, so an enzyme that gets proteins and chopped them to pieces. And he put that together with the other cells, and there was transformation. So if they inject on the mouse would die here. So the transforming principle is not protein. They did the same with RNA by adding RNases, and there was still transformation, so it's not RNA. But when they did that with DNAs, no transformation. The bacteria could not become virulent. Therefore, this is the molecule that gives heredity. The basis of heredity is DNA. And it is not to be taken lightly. That was a massive finding as well. Because at the time, chemists were starting to become interested about biology because they could start to look at those molecules, and they were studying what is DNA, how sugars look like, how proteins look like, and how DNA looks like. And they had no idea what all those things were actually doing in the cell. But now they had the one to really go for it. They had to understand DNA. And that's when we get to 1953 for the story that I said in the beginning, where Watson, Crick, Wilkins, and Franklin made the incredible discovery of the structure of DNA. Those three got the Nobel Prize. She unfortunately died before the Nobel Prize was given. Whether she would be given the Nobel Prize, nobody knows. People think not, because she was not given the whole credit. But I think we are setting the record straight nowadays. So these are the four ones that deserve the credit. So now we're not going to be talking about storing anymore, because the timelines now are a bit more fuzzy, as I said. So what is DNA and what does it do? So as you probably have heard of that, but I'm going to start really from the start so we can build up to recombinant DNA technologies. So DNA in the cells, they are arranged generally in structures called chromosomes. When the cell is functioning normal, this is the typical picture of a chromosome that you see in every in the media and everything. This happens only when cells are dividing, otherwise the chromosomes are much more loose. DNA is much more loose inside the cell. So DNA is arranged in chromosomes, and the chromosomes are inside the nucleus of the cells. We have 23 pairs of chromosomes that we understand very well what they are. We can classify them. Many diseases are caused by parts of the chromosomes that are lost, or parts of the chromosomes that are translocated between one part and the other. For example, Down syndrome is when the third 21st chromosome is triplicated, so that's when you have Down syndrome. And we talk always about chromosome pairs, because here we have the good and old Elementen from Mendel, which is one from the mother, one from the father. This is the particle that Mendel was saying. And the chromosome number can vary massively in nature. Chromosome number doesn't say a thing about complexity, nor genome size, by the way, because you have a giant variation. So flies will have 4 pairs of chromosomes, whales 21 pairs, cows 30 pairs, and there is a butterfly that has 224 pairs of chromosomes. So it just varies massively. People claiming that chromosome number has anything to do with complexity or intelligence or anything, there's nothing there. There's nothing there. It's almost an accident of nature. Evolution have done something and ended up with those number of chromosomes, right? And inside those chromosomes, we have really the unit of heredity, which are called genes, which is this word thrown at the media all the time, and without people properly explaining what they do and what they are and what they aren't. And we're going to try to, at the end of the talk today, to discuss about what genes can and what genes can't do. And a gene this is a very simplistic view of a gene. And you can think of a gene of 2 parts. A regulatory part, which we generally call promoter. Here is just showing like a tiny piece of DNA, but that can be very long, that can be very far away, that can be multiple places in the genome that control a gene. So it's a bit of a mess. It's very complex. And that promoter is the part that will make that gene being expressed and turn into RNA. We're going to talk about that later on. And that part that is expressed is formed by what we call exons and introns. So exon is the part that will become protein. Intron will be taken out, but intron also has some regulatory activity. We're going to talk a bit about that later on. But gene is nothing but a protein-making machine, right? And from a molecular point of view, and this is basically what Watson and Crick found out, the DNA is that beautiful spiral formed of 2 strands of DNA linked to each other in the center, and that linkage occurs via nucleotides. And there are 4 types of nucleotides only on DNA. So the adenine, the thymine, cytosine, and guanine, A, C, T, G, that you see everywhere, right? And chemically, they're very simple. This is the whole chemical of the structure of DNA, and it's linked on a vertical side by this a phosphate group bound to the sugar, and the A's always linked to T's, and C's always linked to G's. So when you read one strand of DNA, you know what happens on the other. And that was the crucial finding of Watson and Crick, because now, and that is probably the biggest understatement in science, they have one sentence in their paper that says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So because the pairing is always specific, if you open up the two strands and copy the other one, because you know what's going to be the pairing. Where there is an A, it's going to be a T. Where there is a C, it's going to be a G. You can copy one, you can copy the other, boom, you have two copies. And by looking at the structure, they could deduce that. And that's exactly how it happens. There are multiple details there, but looking at the structure, you know how DNA is replicated. And it's not an understatement to say the impact that paper had, because now we understand DNA, how DNA works. So we know how the DNA molecule looks like. So now we need to understand how biology works coming from the DNA. And that paper was literally the rise of molecular biology. And one of the first things that came up just after that paper was Francis Crick, the same guy of the DNA. He proposed a thing called the central dogma of molecular biology, which explains what DNA do. So DNA can be replicated by the process of replication. So you can make multiple copies of DNA, cells can divide, and reproduction can happen, and etc., and so on and so forth. DNA is turned into RNA. RNA, it's very similar to DNA, but it's a single strand only. It does not have the double strand. And the other change is that instead of having T's, it has U's. So instead of having thymines, it has uracils. And that RNA will be turned into protein. So every 3 base pairs here on the RNA codes for a specific amino acid that you can make a whole protein. And protein is the real effector of function in cells. Proteins will be the enzymes that will metabolize everything you eat, will be the enzymes that will make lipids, make sugars, make DNA molecules, make the individual components of DNA molecules, the neurotransmitters in your brain, will be the ones that will drive what organelles are doing inside your cell, and will be also lots of structural proteins that will give each cell in your body its shape and its function. So DNA, it's primarily there to make protein. Obviously, biology is not that simple. There's a lot of RNAs that do a lot of things, but let's just simplify here. The main function of DNA is to make proteins, right? But now, so that was proposed by Watson Crick in 1958, the central dogma of molecular biology. So now we need to unlock the mysteries of the DNA. And for that, we need to understand and find ways to manipulate genetic information. At that time, it was not very easy to manipulate genetic information. And that was the birth of recombinant DNA technology, which is a very wide name that encompasses a lot of things. And to give this lecture, I had to think, how am I going to make recombinant DNA technology something that makes sense and it's not just a bunch of techniques thrown together? So I have to excuse myself, I made a big oversimplification of recombinant DNA technology, but I think that's I'm proud of it. It's a good way to look at it. So to do recombinant DNA technology, you basically have to cut and paste DNA. And to do that, you need 4 functions. You need to be able to read the DNA. You need to find ways that you can read those genes and see what's in those genes. After you know what's in those genes, you're able to write them down, make copies of it. So after writing those copies, you can cut and paste it into a carrier. And the reason that the carrier here is important is that chromosomes are gigantic. They are impossible to handle in a lab. For you to be able to study biology, you need tools that are easy to assess. And working with full chromosomes and full gigantic pieces of DNA is not very easy, not even today. Imagine in the 60s. So they had to find ways of having a carrier, like a vehicle to make things easier. We're going to get there. There was plenty, and we're going to have a throwback to the beginning of the lecture as well. So then, let's start by reading. So read DNA was invented in 1977, a method called Sanger sequencing, developed by Fred Sanger. It is a very simple method. The idea is beautifully simple, and it was used for a long time. We effectively just in 2005 or 2006 stopped using Sanger sequencing as the single only sequencing technique that existed. So it's extremely powerful. And so Sanger discovered that in 1977, got a Nobel Prize 3 years later for it. And the interesting thing about Frederick Sanger is that he used to work alone in the lab, himself doing the experiments. And that was his second Nobel Prize, because he got another one in 1958 by learning how to sequence proteins. So I worked in the same institute as he did. So the guy is a legend. Everyone says that the guy is a legend. So interesting guy, very, very, very smart. So then you could read the DNA. Now you need to write it. So you need to make multiple copies of it so you're able to manipulate it in a different way. And to be able to write and modify DNA, two techniques were important. So first of them was polymerase chain reaction, or PCR. If you're not living under a rock in the last 5 years, you've heard of those three letters, PCR, which is the basis of the COVID test that allows us to diagnose if people had COVID. And what PCR does is that it gets one piece of DNA that is very rare and make millions of copies of it. And this is a great way for you. If you have to copy and paste DNA, you cannot have one molecule and one molecule because it's going to be super complicated. So we start with millions of molecules and try to do it from millions of molecules onwards. And you could not only copy DNA, and there was another technique that is called site-directed mutagenesis that was developed, which is based on PCR, that not only you can make copies of that piece of the DNA, but you can change it as well to whatever you want. And those two techniques together gave the Nobel Prize to Kary Mullis and Michael Smith in 1993. So Michael Smith for the site-directed mutagenesis and Kary Mullis for PCR. By the way, if you want to hear a story about an interesting guy in science, read about Kary Mullis. He's a character. Surfer, like super crazy guy, crazy ideas. He's an interesting character. Then you could read the DNA, you could write it via PCR and change it the way you want, and you needed a carrier. So what can you use as a simple way of carrier? And for that we used something from bacteria. As you will see, lots of the stuff in molecular biology depend on bacteria, including the latest developments. Bacteria is an incredible resource for tools. Bacteria are very smart organisms despite being tiny and ignored and hated by everyone. They have very incredible things happen inside of them. So bacteria have two types of genomes. They have their chromosomal DNA, which is very large, but they have also these things called plasmids, which are extra chromosomal DNA. And those are very small. We're talking here about 5,000 nucleotides, while a genome can be millions, billions, trillions of nucleotides. So they are very small, they're circular, you can make multiple copies of them, you can read them very easily. And one interesting thing about plasmids is that plasmid was effectively the transforming principle for growths, because what was transferred between the bacteria, the non-pathogenic bacteria to the pathogenic bacteria, the pathogenic to the non-pathogenic bacteria in that experiment with Streptococcus was a plasmid, because there was no way that you could transfer a whole chromosome. It would not work, the experiment. So he was very lucky that on that specific bacteria, the virulence, the capacity to kill the mouse, was encoded in an extra chromosomal piece of DNA called plasmid, which can be easily transferred between bacteria. So serendipity in science, that's the rule. So that's an interesting story as well. So then you just had to cut and paste. So what can you do to cut? So to cut, you use scissors. And once again, bacteria. In bacteria, we discovered one thing called restriction enzymes, and restriction enzymes are how bacteria defend themselves from foreign DNA coming inside of them. And those are enzymes that recognize specific sequences and chop them. And these people got the Nobel Prize in physiology in 1978, and they discovered hundreds of restriction enzymes. So you have a whole plethora of different sequences that you could cut and paste between the different places. And so you could just read the sequence and say, which one of the enzymes I can use here? I can use this enzyme and that enzyme. It was a bit of a puzzle. I worked for 4 years in Brazil in a company where my work was just do that, cut and paste DNA. And that was a very fun puzzle to do. And so you had to cut the DNA, but you had to ligate it back together, to glue it back together, and that was done via an enzyme called DNA ligase. Nobody got the Nobel Prize for that one, because 5 different groups published it at the same year, in the same one after the other. But basically what DNA ligase does is get those DNAs which are far away from each other and just mediate the ligation of the DNA, and you're able to reconstitute your plasmid again with the piece of DNA that you're interested in, and you're able to study whatever you want with it. So far, everything I'm saying here looks like a very niche thing for a bunch of us geeks in the lab doing fun stuff. But why recombinant DNA technology became so revolutionary? So what's so revolutionary about these fun copy and pasting of DNA here, which looks very niche? So first and foremost, and that's something that I'm going to speak a few times during the lecture here, is the importance of basic science. So the first and most important thing of recombinant DNA technology is that recombinant DNA technology allows us to look at biology and life itself to a level that we couldn't do before. And here we are talking the difference between looking at something from the top of the building and having a magnifying glass on the bottom, looking at an ant on the top of the building and looking to an ant with a magnifying glass close to the ant. That's the difference in terms of magnitude, how much deeper we could understand biology. And I'm going to just give a few examples here, because that could be a 5-hour lecture about that, because that's what I love, that's what I do, that understanding biology using molecular biology. And the first of them are genomes, right? This is obvious. So molecular biology and recombinant DNA technology gave us the possibility to sequence full genomes, not only ours, so our genome was fully sequenced in 2000, but also to sequence the genome of multiple species. Nowadays, we know the sequence of the genome of thousands, if not hundreds of thousands of species. We have sequenced millions of humans. And we know nowadays things about our own biology and our own history that 20 years ago was unimaginable. Things, for example, we are able to sequence people from all over the world, and we can now very easily trace how populations migrate out of Africa and went and spread throughout the whole world. Genomics has given us the possibility to debunk crazy ideas that there are different races in the human race. We are a single race, and there are larger variations between the so-called races than between the races that we call races. So we are a single race, and that is proven 1,000,000 times over by genomics, right? But also understand different populations of different organisms and biology and the beauty of biology, and will help us to tackle big things in the future like climate change and etc. But also, and this is something very close to my heart, that molecular biology and recombinant DNA technology allows us to look at proteins inside the cells and understand what they are doing. So on recombinant DNA technology, you're able to take a gene of interest and put that gene together with a tracer molecule, it's a fluorescent molecule, and put that back inside the cell, and you can look at the cell and see where that protein is and what that protein is doing. And you can make movies out of it and realize when the protein is going from one place of the cell to the other. And here is just a picture of the cellular cytoskeleton with microtubules in yellow and the actin cytoskeleton in blue. And nowadays with molecular biology, we can make cells all colorful and image them and have incredible movies and see all these structures forming and polymerizing and depolymerizing and making the cell move, making the cell divide, and so on and so forth. And this is only possible because of molecular biology. And also, we can understand protein structure as Watson and Crick taught us. Form many times tell function very clearly. And molecular biology and structural biology allows you to produce proteins or protein complexes and make and look at their structure and understand where every single atom of that protein is and what is the shape of that protein and understand their function. And this is absolutely essential, for example, for to make medicines and etc. Because you can look at how a molecule works, and so you can design drugs for it or understand how drugs work. So absolutely revolutionary. And so here it's mostly from a basic perspective, but there are incredibly useful applications for recombinant DNA technology as well, right? So diagnostics, as we already discussed, the COVID PCR, thank molecular biology for it. Genetic testing, nowadays you can see if someone, you can diagnose the disease of someone. If that's a genetic disease, you can sequence their DNA and see, okay, you have that disease because that's a mutation that you have, and that's the reason that you have that symptom. Or if you have a cancer, for example, you can say this cancer is because of this specific mutation, therefore the therapy better to use is this one. So it has gone pretty far. But undoubtedly, the most impactful use of recombinant DNA technology is in therapeutic products. So insulin, growth hormones, therapeutic antibodies, all is produced via recombinant DNA technology. You modify organisms to produce that for you, and you develop whole industrial processes to produce therapeutics. By far, the most valuable contribution, monetary value contribution of recombinant DNA technology. Millions, billions, if not trillions of dollars on recombinant DNA has been therapeutics for used from recombinant DNA. And you have also uses in agriculture, right? It's a bit of a controversial field. You can do genetically modified organisms, which is a discussion that we can have, but also, regardless of genetic modification of organisms, molecular biology have helped us on regular breeding, plant breeding, to make plant breeding more efficient, to make plant breeding faster. So recombinant DNA technology is not only to genetically modify crops, but to make them better by conventional methods. So we can discuss that later on if you're interested. But the list goes on and on. So biofuels, there are lots of companies in the world. I recall when I was working in Brazil in a company, the sister company on our side, there was a whole research program on trying to make sugarcane produce more sugar to make more fuel for biofuel. So there is a whole industry on that. CO2 capture, there are people genetically modifying algae for them to become more effective at capturing CO2. There are projects on bioremediation as well. Bioremediation is, for example, when you have an oil spill in the ocean, and there are groups working on bacteria that can eat oil, and they are able to trying to engineer them and able to control them, because you cannot just release something, you have to control them. They're finding ways that you can do bioremediation via recombinant DNA technology using genetically modified bacteria. And obviously, and that's probably the reason that you're here, genome editing. Recombinant DNA technology allows us to do genome editing. However, if you look at the history of the field and imagine that we could be doing most of those things 25 years ago pretty well, why at that time, 25 years ago, you would hardly hear about genetic engineering of humans? Because at that time, I mean, you would hear that just on movies and things, because what was wrong there? And there are a very specific reason for that, and I'm going to try to explain that to you. What is the challenge of genome editing using what I'm calling here old school recombinant DNA technology, where you have that piece of the gene that you put that on a plasmid, and you have the recombinant DNA, and you want to put that DNA inside the genome to correct a mutation or to produce something interesting or to disrupt a gene that you don't want? What's the challenge of that? So we do that all the time, of getting the recombinant DNA on a plasmid and throw that inside an organism. That's basically things we do on a daily basis in the lab. But when we do that, there are three things that can happen when you throw a piece of DNA, plasmid DNA, inside a cell. And that is true for most organisms here, right? The first and the most common thing is that that plasmid will stay inside the cell for a transient period of time. So it will stay there for about 2 weeks, 3 weeks maximum, because that piece of DNA do not have the signals on it for it to be able to multiply inside any place but a bacteria. So that one, when the cell divides, it's just going to be in one cell only, but also sometimes it will just escape and be degraded, so it just doesn't last very long. For what we do in the lab, it's perfect. We don't need it for more than a few days, and we're fine with that. Very interesting tool, but absolutely useless for long-term experiments, right? But what can happen, and that happens sometimes, is that piece of DNA just integrates into your genome randomly. And that's something that you don't want, right? Especially for therapeutic, you don't want just something entering in the part of your DNA. And sometimes this can break apart into small pieces, and each piece goes in a different place of the genome. So it can be pretty messy. And that is the biggest problem with genetically modified crops. Because genetically modified crops, the old school ones, they're all done like that. So in many cases, you end up with pieces of DNA kind of scattered throughout the genome. Plants can cope really well with messy genomes. Superior organisms, mammalian cells, a bit less. So it can happen, but it also can have specific targeting. It's possible to specifically target. The problem of that is that this is extremely, extremely rare. We do that nowadays with model organisms such as mice. We can do with mice, but you have to add so many other things inside that plasmid to try to target to the same place. So that you have selections and counter selections and markers and these and that, which is something that on a therapeutic perspective, it's completely unacceptable. You don't want an antibiotics resistance gene inserted into your genome, right? There is absolutely no way of that. And and that is doable in mouse cells, but in humans it's not possible. I mean, even in cell lines in the lab, we just can't do that. So that is the problem of old school recombinant DNA technology. It's just not suitable for genetic engineering, for precise genetic engineering. It's a very crude, bazooka-like technique that is a bit too much. It just doesn't fit the finesse you need for therapy. But things started to change a bit. And there were, since 2002, some tools that could possibly do site-specific genome targeting. So the first one was called zinc finger nucleases. Those were discovered in 2002. They are very fiddly, very difficult to engineer. You have to test many of them to see the one that is specific, that is correct. So kind of ended up dying out. And in 2011, something a bit simpler came about called TALENs, which is pretty all right to do. But unfortunately, 2 years later, CRISPR came up. And basically, CRISPR killed everything else because it's so much simpler, so much more powerful that it's a no-brainer. You just don't do that anymore. So that's the reason that we're going to be talking until the end of our talk here about CRISPR, because CRISPR is the future, right? And CRISPR, once again, is the power of basic science. CRISPR just exists because people look at the government that do not want to fund basic science and say, I don't care, I still gonna do it. And these people had exactly what they did. So CRISPR is the result of pieces of the puzzle coming together. CRISPR as a gene editing tool just became obvious as a gene editing tool when the last piece of the puzzle came together. Before that, people had no idea that that was possible to use that. People were just studying CRISPR because it was something interesting happening in bacteria. And those two ladies, Emmanuelle Charpentier and Jennifer Doudna, they were the ones who got the very last pieces of the puzzle here. And they got the Nobel Prize well-deservedly in 2020. It was the first ever chemistry Nobel Prize given only to women. A very important moment for science and an incredible moment for the field as well. So the paper that they published has a lot of very interesting things about it. So the paper that they published first, it was a paper only on the basics of CRISPR, understanding the principle of CRISPR, nothing to do with genome engineering. But they had one sentence in the paper saying that our study highlights the potential to exploit the system for RNA programmable genome editing. And the paper is a basic science paper, it's a basic scientific paper, and that was so revolutionary when you read it and when you realize the potential of that paper was, from submission to acceptance in Science, in the journal Science, was 12 days. This is unheard of. This is very rare to happen. Generally, a paper in Science, you have 1 or 2 years of revisions. So a paper go from, it's really rare. And they have indeed revolutionized the world on that because they have found a way to make CRISPR very simple. You can do a full genome engineering by using one single plasmid. You use one plasmid that expresses the system and basically just does your genomic engineering. So what is CRISPR then? So in the bacteria, and that was what people have been studying for 20-something years, is that CRISPR is a bacterial immune system. So what CRISPR does inside the cell is that you have a piece of DNA from viruses that infect bacteria, which are called bacteriophages. And when that virus infects the bacteria cuts that DNA. Remember restriction enzymes? Remember restriction enzymes there long ago that I said, the ones that cut the DNA? And that DNA ends up inserting into the genome of the bacteria in a very specific place, like a sign for immunity. And what that does is that generates this CRISPR complex, which basically targets that piece of DNA in the beginning to eat other DNAs from other phages that are infecting it. So it works like an antibody. You'll be infected once, and you will remember that, but when next time that infection happens, you're going to have the tools to chop it off, the DNA that is coming inside of you. And what they have done, this is in bacteria is far more complex. What Emmanuel Charpentier and Jennifer Doudna did is that they discovered how the system worked and simplified it. And the system can be seen as a two-piece system now. They could simplify it in a two-piece system, where you have one enzyme here, which is called Cas9, which is this bean-shaped thing here, and you have what is called a guide RNA. And what the guide RNA does, as the name says, it guides that enzyme to the right place on the DNA, because remember, there is pairing of bases. They are always A with T, C with G. So the guide RNA takes that enzyme to the right place at the DNA, and the enzyme can cut the DNA very precisely at that spot. And when the DNA is cut, there is a whole system inside a cell called DNA repair mechanism, a bunch of mechanisms called DNA repair mechanisms that are triggered. When your chromosome breaks, the cell can detect that and say, something is wrong. We have to repair that as fast as possible. And what happens is that you can take advantage of that system to actually change whatever was on that cut. You can add different things. You can delete things. You can do different types of trickeries with that. And the system is really clean. I mean, it's not 100%, but it's really clean and very easy. That has really become the gold standard in biology nowadays. And because the system is so simple, there are a bunch of variants. Every 6 months there is a new paper with a new CRISPR variant that instead of cutting the DNA, there is a deaminase enzyme that will change a base, will change a specific base pair, another one that will bring other different insertions with transposases, and other things that will stop the DNA, and the list goes on and on and on and on. There are easily 25 different types of Cas9 of CRISPR nowadays. Easily. And it's just not worthwhile going there. But what it can do, it's the same base. Something takes it to the DNA, and there is another enzyme that can cut, can change, can insert, can remove, can stimulate, can repress, name it. CRISPR can do it. It's ridiculous what it can do, and with its simplicity. And what can we do with CRISPR? And that's more of a curiosity, because when I was preparing that lecture, I started to Google and look for reviews for uses of CRISPR for different fields. And I realized that every field loves a wheel. Every single review about CRISPR, there is a wheel of uses of CRISPR. So here is a wheel of uses for potatoes. People who grow potatoes and want to do genetic engineering of potatoes. Here's people who work with fungus. Here's for cancer research. Here's for biophysics. Here is a general one, and that one is for hematology. So every single one has a different wheel, and they basically talk about the same things, but with their own view of things. So I decided to just simplify it here. Basically, with CRISPR, you have 3 things that you can do, which are the same as we could do with recombinant DNA technology, but now with a much bigger finesse. So the most important one, basic science. We can understand biology, we can understand life, understand how systems work. And that, because, as I said, has become using CRISPR is the gold standard in science nowadays. We use CRISPR all the time in the lab. Just to imagine that's something that was invented 10 years ago, 11 years ago, it's already every single lab will have CRISPR running. It's really incredibly simple and incredibly powerful. And we can do genome editing, but also it can be used in bioproduction, because now we can tweak the bioproduction in a much better way than we could do before, right? And now let's talk about genome editing, which is the important part of the lecture, and the one that I am the least knowledgeable about because I'm no bioethicist. So I'm going to try to look at that from a technical perspective. Okay? So, first of all, an important distinction that has to be done when we talk about genetic engineering for therapeutic purposes is that there are two types of editing or engineering you can do. One is called germline editing, and the other one is called somatic editing. So, germline gene therapy or germline gene editing, it's when you edit the embryo, which means that that embryo will be edited and every single cell on the adult will be edited as well. And that adult, if he or she or they reproduce, it will also, their sons and daughters will have that change as well. So this is a long-lasting thing that can change populations, and very controversial, extremely controversial. And the other type, which is somatic gene therapy, and that's much more realistic and much more sought after by most of industry and scientists. The somatic gene therapy is when you just change the cells which are sick, which means that, and you're going to do that when the person is already an adult, when the person is already born. The person is born and you realize that they have a disease in an organ, so you're going to remove some cells, you're going to edit them, you're going to put them back, and those cells will be changed. And that lecture is in a great time because effectively last year, the end of last year, we had the first ever CRISPR-based therapy approved by the UK government and the FDA. It was in October and November, I guess, the approval dates. And this is gene therapy for sickle cell anemia. So what they do is they extract the stem cells from patients. So by the way, sickle cell anemia is a terrible disease, and kids are extremely sick. They have seizures. It's a terrible disease. And the problem is their hemoglobin, which is that protein that makes your blood red and transports oxygen through your blood. That enzyme, that protein is defective. That's the problem with a sickle cell anemia. So what they do then, they remove the stem cells from the bone marrow from patients. So your bone marrow is the place where all your blood cells are produced in your bones, in your large bones. So they extract the stem cells, which are the cells that will produce more blood cells. Extract those cells. They change it in the lab, so it's really something done in the lab. And after that, they have to undergo massive chemotherapy on those patients to basically kill all their stem cells in their bone marrows, and they reimplant the genome-edited cells. And on their initial trial, they had 91% of people with very good outcomes. So it's not bad. A problem costs $2 million, because it's extremely difficult, because here you change it, but you have to do genome sequencing to ensure that nothing is wrong, and a lot of controls. This has to be done in extremely sterile environments. It's a really complex thing, but it's incredible that in 11 years, something came from a basic discovery to a therapy. This is incredible. But unfortunately, the real discussion in media, it's not about that. The discussion in media is about that. About new CRISPR babies, engineering humans. Even the Spectator was a bit too extreme, saying that eugenics is back. So this has taken an incredible run in media and in popular minds about, we're going to have superhumans in the future and everything. But in the scientific community, that was just, a crazy idea. I mean, most scientists you ask, you say, No, you don't do it. You don't do it. I mean, obviously we're not going to be doing that. Obviously we're not going to be doing that. Unfortunately, someone did do it. And that was this guy called He Jiankui from China. He was not a cell biologist, he was a biophysicist. And this guy just ran for fame and did actually 3 children were born with where he edited the gene called CCR5, and there is some evidence that people without that gene are immune to HIV. So that was the idea. He would do babies which were immune to HIV. But the work he has done from a purely scientific point of view was terrible, and ethically speaking, even worse. And if you want to know details about that story, there is this book called CRISPR People. I read this whole book for this lecture. It was super fun, a bit depressing in some points. And the author for that, he really defines the view of most of the scientific community of what that experiment was. It was criminally reckless, grossly premature, and deeply unethical.
I think that Chinese guy gave a lecture here in Helsinki.
You're joking, really? He got arrested. He just was released from jail because he broke every single. He broke so many laws. You know, He was a terrible scientist and a crook legally as well, and in ethics as well. But I didn't know.
He managed to help some mother with her unborn child. That the child was born in a better condition than.
Actually, that's not the case. That's really not.
It is different.
Yeah, I mean, it is really not the case. That's a really good point. That's good that you asked that. So what he did was those 2 kids, Lulu and Nana. So first of all, so the reason that it's scientifically so terrible what he did is that the deletion of CCR5 provides resistance against 1 strain of HIV. There are the 2 strains which are not, which CCR5 deletion do not provide resistance. And one of the children had one of the copies of the CCR5 still intact. So that kid is genetically modified, and the genetic modification, but it's not immune to HIV at all. And what we know that causes resistance, and there are 2 cases in the literature of that only. What we know that causes resistance is a 23 base pair deletion. One of the kids had a 15 base pair deletion, the other one had a 1 base pair deletion. We don't know what those deletions do. We don't know if they provide resistance the way it's suspected to be. So, regardless of that, scientifically it was terrible what he did. It's very likely that those kids are not immune to HIV. And yeah, there are so many problems. I mean, we can discuss that later on if you want, but it's scientifically terrible. I mean, the scientific basis to do that is also very weak. You know, there are not a lot of. We can discuss the details on that later on after the talk. So I will gladly talk about it. If you want to read more, read that book. It's a very interesting book. So to finish, then let's address one question. Let's put ethical concerns to the side for a bit, and let's ask: can we achieve what these people are claiming on those crazy magazine covers, right? Like they want to born a kid which is going to be a sprinter, will have perfect pitch, will have no baldness, will have a high IQ, will have a low risk of Alzheimer's, breast cancer and strokes, a 20/20 vision. Let's imagine that can we do that? The question is, can we do that? What are the technical challenges of trying to achieve that? So, the first of them is what we already discussed. CRISPR is very specific, but it has some off-target effects. So sometimes CRISPR can cause mutation in other places. It's a bit stochastic. That's it. Sometimes it's 99% of it's precise, but sometimes something else can happen. And the way you discover if something happens is by doing sequencing the genome of that cell. While in a therapeutic process, you're able to sequence the cells because you have multiple cells that you have done. You have done the modification. You can expand those cells to thousands, hundreds of thousands, millions of cells, and you can sequence the genome very carefully and look for things into big detail. On an embryo, you're going to have one cell. So you divide in 2 or 4, and you pluck one cell out, and that cell you're going to sequence. Sequencing the genome of one cell to the depth that is required to ensure 100% that nothing else went wrong, we're not there yet. We're going to get there, but we're not there yet, for sure. Another challenge is physiology is very complicated. This is a metabolic map. You cannot see the lines, you cannot see anything because it's so complex, but this is a map that shows every single metabolic reaction that happens in your body, and it looks something, this is just an overview, it looks something like that, a chemical compound, and here in blue it's an enzyme that is defined by a gene. And that enzyme has a level of expression, is affected by environmental factors, and etc., and so on and so forth, and we do not understand what happens when you change one of those, because you know that people who are older have less of that enzyme and they are less propense, for example, to have Alzheimer. We don't know what that happens when the embryo is developing. We don't know if that happens if that person comes from Brazil or from China, because we just don't know. Physiology is extremely complex. And when we look at physiology, we look at a specific window of time and a specific set of people. We cannot make generalizations in biology. It's really complex. So sometimes despite some claims, physiology is very complex. And even if you would ignore that, there are bigger problems, which is, despite what Mendel said, the majority of the characteristics that people want to change, they are polygenic. They are the result of many, many, many places in your genome, many, many, many genes. The latest research that study height in humans have mapped that there are 10,000 regions in our genome that determine how high we're going to be. And those 10,000 regions are responsible for 40% of the difference in height between people. The other 60, environment. And that is the key point that I think those dystopian ideas of improving humans on traits which are not disease-linked or monogenic or very classic things, like making people smarter or faster or taller or more beautiful and etc. Because genetics is not deterministic. We are the result of our genes plus our environment and our lifestyle. That defines what we are. So the idea in media that they found the gene for that. There's always like that. They found the gene for obesity. They found the gene for that. When I remember I think the latest one that they found on obesity, we found the gene for obesity. And when you go down to supplementary table 23, line 10,570, it says that the gene is responsible for 8% of the variation of weight. So yeah, it's 8% only. The other 92% is how much you eat and how much you exercise. You can change the 8%. Are you sure that the person will be obese or not? No, right? So if you have friends who are billionaires and are thinking to make their sons or daughters perfect, and if you have to worry about this dystopian future of genetically engineered humans, either by improving them on because you're a billionaire and you want to have a perfect kid, or a crazy government that wants to make armies of people, my message to these people is, you already lost your boat. We are already engineering humans. And there are much cheaper ways to engineer how humans are. And for good or for bad. Give them good education or shove them with fake news. It's a much more effective way of engineering humans, unfortunately. And that's what I had to say. I'm just going to leave the last slide here, which is what Jennifer Doudna said about engineering babies, that at the end, we want to take the time to think about it. We need to understand biology better. We need to think about it. And that's what I had to say. Thank you. And I would love to hear comments, ideas.
Thank you, Professor Almeida-Souza. We have now some 25 minutes or something like that to have a discussion, and this was very interesting representation, starting from basic research and showing at the end the Pandora box of applications.
Absolutely.
Are there now some spontaneous questions to start? Please say your name to
Okay, good. Yeah, I'm Simo Salo. Isn't it so that there's still a risk if we have to touch the genius of a human that the possible problems will come up after several generations? So we really don't know when will that happen.
Yes, that's absolutely possible. I left it out here because I thought it would be too complicated. Yeah. There is one thing in genetics called epigenetics, which are modifications in the DNA that do not change the code, but it changes the behavior of DNA. And there is, for example, a typical case of epigenetics was a case studied in the Netherlands, where the kids who were born from mothers at a specific time of the history where there was a famine in the Netherlands, I can't recall the details, the kids who were born from that, they had a specific characteristic that were passed to their to the grandchild, and you could detect the effects on the changes, on the epigenetic changes. The DNA sequence was not different than most. It was just an epigenetic change that was passed to two second generations, and that has effects in physiology. And obviously, as I said, the modification might not make a difference when the environment is the one that we are now. But if that person suddenly changes to a very polluted city, or become an athlete, or become a smoker, you can start to see crazy things. But physiology is complicated. Genes are not deterministic. It's difficult to predict what's going to happen. That is a big challenge.
Yeah, now we are. The environment is changing, but because of climate change and so on. So does this technology helps us to adapt the changing environment? Because it may be that it happens quite in a speedy way.
Yes.
So the evolution is cannot keep on that. So does this gives us some ways to adapt?
I think for us humans, no. I think we are better off changing what we do rather than what we are. So I don't think because we don't know what's going to be the effects of of climate change on us in our physiology? Another big question here. Obviously, I don't think that in European countries that's not going to become a big issue. I'm worried about the countries close to the equator where temperatures will suddenly become 50, 60 degrees on a common basis, and I'm not sure if human physiology is ready for it. But I think genetic engineering can help us mitigate the effects via biotechnology applications, CO2 capture, biofuels, and so on and so forth. But us, no, we have to change what we do, not what we are.
Yes, I did not meant that human beings.
Yeah.
But
there is one thing that is definitely going to make a difference is on crops. We might need to adapt some crops really fast to extreme environments to be able to not lose producing land if things turn too bad, and genetic engineering can help us do that. So plants are much more amenable to, they are much more flexible in terms of modifications. Plants can have their genome completely messed up. Actually, most of the plants we eat, their genomes are completely messed up. Like, yeah.
My name is Juha Someland. I have one question about the future. Are there applications of artificial intelligence to manage the complexity of the human processes?
Absolutely.
And what they could be?
So there is already artificial intelligence, machine learning is part of biology nowadays, and the whole question is. We all know that machine learning require very large data sets and very well trained data sets to be able to come up with something meaningful. And the real challenge for biologists nowadays is to have that data well annotated. So we still need to understand some things to be able to feed into the artificial intelligence and come in, but there's a lot of smaller things that are being done. And I believe that artificial intelligence will be able to solve a lot of the things that we just can't figure it out because we are just too limited. But we need the data, well annotated, curated, and that requires people, time, effort, and funding, which does not happen. Yeah.
Thank you. Thank you. You mentioned before we started something about your lecture about creative thinking in science or something like that.
Yeah.
I think that this is a good area to let us be not only engineers but also some kind of designers maybe and artists. But the freedom of art is maybe dangerous. And you mentioned somewhere in the slides about law. How you see the role of legislation in national level and in European level and in general need be an international law about regulate this development?
There is an incredible amount of discussion about that. Actually, I have one slide here that tells it. So this is not legislation. This is what the World Medical Association talks about study in humans. And this is not in law yet, but basically the most important points here is medical research involving human subjects may only be conducted if the importance of the objective outweighs the risk and burdens to research subject. All research involving human subjects must be preceded by careful assessment of predictable risks and burdens to the individuals and groups involved in the research in comparison with perceivable benefits. So the word predictable here basically throws genetic engineering of humans out of the window very fast. And the fact that we do not understand another part, which is the research, the objective outweighs the risks and burdens. These are questions that this is not law yet. Obviously, what European Union and the United States and NIH are looking at, obviously, it's based on these types of already assessments by the medical community. And I think that there will be, if we want ever to do that, the discussion will take a long time, because every single way that we have done research and medicine in a responsible way, they throw out of the window CRISPR really fast. So medicine is used to do in a certain way, and that is a big issue. Yeah.
How about food production then? Do you think that the risks have been, been overcome already, that we are ready to do food production with these kind of herbs?
So I think we are, and the reason that I say that is because you have to consider what we've been doing for many years. So plant breeding, actually I have a master's in plant breeding. Plant breeding is a mess. You basically get the genome of cells and you cross species which are different with each other, and their genome starts to break apart. For example, the chromosome numbers, you remember there, right? We are n equals 23 pairs. If you get a genome of sugarcane, for example, is n equals 110 minus plus and minus 50 or 15 or something like that. It's a mess. So a lot of the plants we do nowadays, they are the result of very crude manipulations. One way that we can do plant breeding nowadays, like improve plants, it's by throwing a mutagenic substance on top of a seed that basically will make a bunch of random mutations, and we grow them and we select the ones that are better. Do we sequence the genome of those things? No. When we cross a bunch of plants together and make new foods, do we sequence, do we check if they're okay? No. But when you get a genetic manipulation and you do that on one specific and you change 2 base pairs on a plant that we already know, that is forbidden. I think people have to take things with a pinch of salt here. We've been messing about with plants for a long time, and there are very specific ways to test if plants are safe. We should do them. We should be strict. It shouldn't be just keep doing it. And yeah, the technology is there. It's possible, and I think we should be doing it. I'm big favor of that.
If we can manipulate food plants with this technology, what about forests and trees and everything? How much is developed this kind of study in
I think
forest economy?
In the forest economy, so it could be. I recall. I mean, I just for the full disclosure here, I worked in a company for 4 years who used to do genetic modified plants, and one of the ideas of the company was to do eucalyptus for paper production, and one of the ideas was to reduce the amount of lignin on eucalyptus, because you have in the plant you have lignin and cellulose. Cellulose makes paper. Lignin is just a pain to get rid of. So the idea was to make eucalyptus with less lignin, so you could reduce the amount of chemical waste that you require to do paper. And I'm not sure if that actually ended up on something. I left the company later on, but I think there are possibilities there. But I would be much more careful. I would not be comfortable on adding genetically engineered plants into an environment where there are natural populations of that plant close to it. that shouldn't happen. You should be sure to contain them into environments that will not cross with natural plants. We should not contaminate the environment more. Let's stop doing that. So,
how can you capture more carbon dioxide with these techniques?
you need to use, so there is a very specific chemical reaction that captures carbon dioxide. There's a thing called C3 and C4 plants. Some plants that capture carbon dioxide during the day, other plants that capture carbon dioxide throughout the whole day. And we understand the physiology of that. It's a bunch of chemical reactions. If you're able to make that even more efficient in a very simple organism, you can have vats of, for example, algae or bacteria that can be basically, you feed carbon dioxide into them, and they transform the carbon dioxide into sugars, and you brew beer or something out of it. I don't know. Yeah, that is basically what people have been doing. We've been doing with algae. But the idea is there are we understand very well mechanism of carbon conversion into sugars. It can be done. Whether it's economically viable, that's a different story.
You mentioned about the deterministic questions in biology and what is indeterministic and
Not deterministic.
then not deterministic, yes. And the idea is that genes are transmitting information, and information theory probably has important connection to your studies, and it is combined to probability calculus. And what about the level of using probabilistic models in biology? May be quite high.
It's used quite a lot. Gene expression, a lot of gene expression models to understand how multiple genes are expressed throughout different. They use probabilistic model rather than deterministic models, because there is, as we were discussing before, there is quite a lot of noise in how genes are expressed, and there is a bit of stochasticity on how the whole thing works. So you have to use probabilistic models rather than deterministic models to understand some of those processes.
There are so many applications we have also discussed a little bit, but what about biofuels at the practical level? Can we expect that this would change the situation even in economical level, so that the world is changing in that aspect?
Mm-hmm. Yeah. So obviously with electric cars coming into play, biofuels are losing some ground, but obviously they can. I think biofuels have the incredible possibility of being a transition fuel, right? Even if you stop selling electric cars today, there will be still a lot of cars on the street for a long time. And one thing that most people that when I talk about biofuels don't know is that in Brazil, we are 40 years ahead on biofuels. So in Brazil, we have cars which can run on petrol or ethanol in any ratio at any amount for 25 years. For 25 years, you can get to a gas station. The majority of cars in Brazil, you can get to a gas station. There's a pump of ethanol, there's a pump of petrol, and you can just take the one that is cheaper on the day, and you can just fuel your car. And lots of the Brazilian cars run on ethanol, on biofuel. And there is an incredible and vibrant industry on improving, and mostly of it is done in sugarcane. There's an incredible industry there to do it from sugarcane and use some biotechnology on it. I don't think it never really became genetically modified But it's a transition fuel. We cannot ignore it. We cannot ignore it.
We have still some questions.
If you have questions for later, you can find my name and email me. I will answer as well, gladly, no problem.
Maybe one question. This is of course based on natural sciences, but what about humanism, and you mentioned ethics? How much is common work with in your group with humanism and sociology and psychology and other sciences?
For what we do, unfortunately, not enough. I mean, we obviously, when we have to do some experiments with mouse models, my group, we do not work with any animals at all. When people have to do that, you have to go through an ethical committee and look at things. But I think in general, and this is a big shame that I don't think biology, philosophy, and social sciences talk enough. I had the incredible pleasure to work with a philosopher, friend of mine here from Helsinki called Lillian O'Brien. And we wrote an article about kindness in science and how labs should work. And that's something that came in the middle of the pandemic. We knew each other. We started talking about it via Zoom and just wrote an article because that's what we had to do. And I realized that we've been doing biology wrong for a long time. We need more philosophers and social sciences around us to help us to ask the questions we ask in the lab in a different way. I think, yeah, we must put all these people in the same room together. Every biology lab should have a philosopher and a social scientist in the same environment for you to have coffee with them and discuss about your science. I think it would just make biology and probably social sciences and philosophy even more interesting. I think we need more to discuss that. And when it comes to tricky subjects like that, having someone with a view from human sciences to discuss about them, it's crucial, because I got asked about that. My mom asked those things to me. My friends who were not biologists asked those things to me. And I always felt not equipped for it. And by reading that book, I kind of realized how much I needed that. And yeah, we need to talk more.
How big is your group? How many theses are under work now?
Sorry?
How many theses are under work with your students?
Okay, you mean, so I run a research group. We're 6 people in my research group.
Yeah.
And we study cell biology, and I'm there 5 days a week. and I give these a few courses on basic biology, and I run this course on creative scientific thinking, which runs throughout the whole year.
Okay. Thank you for the speech and then about the discussion, and thank you for the auditorium about the questions. And we have now the time quite in hand, so let's give a little bit more applause.
And thanks a lot for the discussion. It was really fun.